So, okay, now are we ready for the next presentation, which is supported by Елта 90 from United Kingdom, Daniel?
Yes, can you hear me?
Hi.
Good afternoon, can you hear me?
Yes, perfectly. We can see you as well. So now we are waiting for your presentation as well. Welcome.
Please confirm that can you see my screen? You can see my screen? Hello, can you see my screen?
No, now we can see you. Yes, now it's okay. Yes, we can see your screen. Thank you.
Okay, so thank you for the invitation and the opportunity to speak, and good afternoon to everybody. My name is Daniel Lieber, and I'm the Business Development Manager for the automation system at Takara Bio Europe. So today I'm going to present our single-cell technology instrumentation for single-cell genomics. And I will just have a kind of a brief introduction about the context. So we work in the field of single-cell biology, which is actually a very old field of research. It's probably more than 500 years old. But of course, over the last century, it has developed and changed drastically and in a considerable way. So from the very old anatomical analysis of single cell, where kind of people were looking at the different cells and looking at the shape and try to differentiate them based on the kind of morphological characteristics, we kind of move in the last century and in the last particularly 10 years into the capability of having a very different and much more detailed analysis of cells with the kind of starting of the single-cell genomic era. It's just over 10 years old field of research. So the very first publication is from 2009, where a single cell was first analyzed in its transcriptome. And from that initial paper, now there are kind of thousands and thousands of publications that have used similar technology or develop of similar technology to study the genomic characteristic of individual cells. And Takara is been kind of involved in this field for a very long time. And the technology that is actually at the base of most of the single-cell work that is done today was developed by Takara more than 20 years ago. It was developed for completely different purposes. It was developed for to do cDNA clones. But the basic of the technology, which is the switching mechanism at the 5' end of the RNA template, it what allows to amplify mRNA from very small input amount that can be compatible for single-cell analysis. So in 2011, Takara Bio released the very first kit on the market to allow to do single-cell RNA-seq based on this technology. This technology have kind of evolved over the years. We are now at the 5th generation kit, where we have made a considerable improvement in sensitivity and to simplify the workflow. But another big development at Takara was in 2015, the acquisition of Wafergen Biosystems, which had developed a technology called the isolate to allow single-cell isolation and processing for sequencing in an automated way and a high throughput. And in 2018, Takara launched kind of the 2nd generation instrument of the isolate. So a step backwards. So why do we want to study single cell and their kind of genomic interpretation of a single cell? Probably a lot of people in the audience are used to type of experiment where you take a sample, which can be a pool of cell, can be from tissue culture, it can be from biopsies, it can be cells that are circulating in the blood. All these cells are taken together, they are mashed up to release the genomic content, and then you can do, for example, qPCR or other type of assays to determine, for example, the expression of specific genes. Of course, once you have a mixed population, it's very difficult to understand, okay, what is the context of this gene that I'm seeing expressed at different level within this sample and comparing, for example, to another condition. So what single cell allows us to do is to separate each of these individual cells and process it individually. And we can do this across multiple samples. And in this way, we can really understand, okay, what is the property of this specific cell? What makes this cell different from a transcriptomic, for example, point of view, but also from a genomic point of view from the other. And then, for example, if you're doing treatments or these samples are subjected to any kind of alteration, how these populations are, for example, altered based on these conditions. So this kind of technology and kind of all the development in molecular biology to be able to analyze these cells at high throughput allow us to have a much deeper understanding of samples of tissues and molecular processes. And these have been applied in the last 10 years to kind of a variety of research fields from kind of oncology, developmental biology, stem cell research, immunology, etc. Basically, in every model system, in every system that is available, single-cell genomics allows to have a very deep understanding and interpretation of these systems. So what is our technology and how can our technology be used to do single-cell genomics? The core of the system and what we use to isolate the cells is the isolate chip, which is a metal alloy chip. It's a kind of a small device. It's 4 centimeter by 4 centimeter. It's a square. And what is inside this chip are 5,184 individual reaction wells. So you can consider these as a kind of a highly complex and highly parallelized multi-well plate. So in each of these chips, we have the same capacity of equivalent of 14,384 well plates. So we have a very high capacity to isolate cells and then do the processing. Of course, the advantage of having all of these in a single device is that we have more reaction that we can do at the same time, but also we have miniaturized the reaction volume. So we can use less reagent to generate the libraries that will then be sequenced on Illumina sequencing machines. Also, what this allows us to do, this kind of in concept simple device of having individual wells that are physically separated, is that we can add reagents into this well relatively easily. You can imagine these wells are really small, so we have kind of developed all the robotics around this, but we can very easily add reagents at different steps to create complex chemistry, to be able to address and develop chemistry to analyze different aspects of each of the cells that we isolate. So what is the Isolate CX system? We have kind of the hardware part of the technology, which is this system that you see here. It's a benchtop device, is roughly 100 centimeter wide, 60 centimeter deep, and 70 centimeter high. And what's inside it is our 2 main components. So we have a dispenser that is capable to dispense in very small volume and to address each of the well of the chip independently. And we use this to dispense the cells. And then we have imaging capability. So we have an internal microscope, fluorescent microscope, that allow us to identify where there is a single cell inside the well, and by using fluorescence, we can also do some other characterization of these cells. The final kind of component of the system, it's again the dispenser that I showed you before, that on top of being able to dispensing cells can also dispense the reagents that are required to generate the libraries ready for sequencing, so we can have an end-to-end library preparation, cell isolation and library preparation instrument to perform single-cell genomics experiment. The system is also composed of different software. So we have the instrument controller software, where we can also have the capability to develop applications. So our user can develop their own scripts for their custom protocols if they want to develop those. We have imaging software analysis that will analyze the images generated by the microscope to identify where there are single cells. And downstream, we have all the software packages that are necessary to analyze the data and to generate kind of allow to help the interpretation of the results. And as part of the system, we also have different chemistry, different kits that are available to perform different type of sequencing experiment, from RNA-seq to DNA-seq to T-cell receptor sequencing and other kind of applications that have been developed either by Takara Bio or by users of our technology. So what the technology does, what makes it unique? So what allows this system to do is to have much better control of your data. Since we can visualize the cells and we can decide which cell to process, we have a lot of control into the experiment and what is generating data. We have the combination of imaging information and the genomic data that are generated from this specific cell. So this information can be correlated. In certain circumstances where people are working with very large cells, one of the advantages of the system is that we don't have cell size limitation or biases, so we can process cells of various size, and we can allow to have a good experimental design by the integration of positive and negative control and the processing of multiple samples to reduce batch effects. The system is also very flexible because it can process multiple samples. So depending on your experiment, you can run between 1 and 8 samples. You can have information on the phenotype and use the phenotypic characterization to select cells. And the system is open to perform multiple applications. I will go very quickly on the kind of the core of the technology. If you have more information, if you require more information, I can go further in this. But fundamentally, as I showed before, we have 2 major components. One is the dispenser, and what the dispenser does is transfer liquid from a source plate, which is a 384 well plate, where samples and controls and in later stages reagents are loaded, and the dispenser will aspirate from this source plate and transfer these samples into the chip. The second kind of fundamental part of the system is the imaging station, which is again is a fully automated chip scanner for the chip that fit into our software to identify the specific wells within the chip that contain a single cell. The dispensing is based on limited dilution, meaning that some wells on the chip will be empty, some wells will contain multiple cells. And what we, the software, want to do is to identify exactly the location where there is a single cell. And all of this is done in an automated fashion. There is the capability to tune the parameters on the specific experimental type, on the specific cells that you're working with. And as I mentioned before, you can use the phenotypic characterization or phenotypic feature of the cells to make a specific selection. So moving in the second part, so what can we do? What type of experiment we can do with the system? So we have a variety of protocols that are available in the system, either in the form of full kits that are kind of end-to-end supported, or we also have different protocols that have been developed either internally by Takara Bio or by some our customer, for example, the ATAC-seq, which was developed by a group at Stanford University in the United States, or cat and tag, which another epigenetic application, which was developed by the Fred Atkinson team. And we also have kind of chemistry that are under development at our end, or again by customers. What I would really like to focus the rest kind of the presentation is technology that are based on our smart technology. This is what I mentioned at the beginning, is the chemistry and the technology that is at the core of our chemistry. And what this allowed to do is a full-length transcriptome analysis. So in this case, we have a kind of a full automated workflow. So everything is done inside the system, and what you get at the end is a sequence-related library that can be sequenced on Illumina sequencers. And what this means is that we can generate sequencing from the entire gene body. Most of single-cell chemistry and technology that are available on the market, they only focus on the 3' end, meaning that they are only useful to identify and call a specific genes, but you lose a lot of information, which is in the rest of the sequencing of the transcript that are expressed in your specific cell and in each individual cells. So what I mean by this is that there is a lot of information that can be gained by looking at the full-length body of the gene. So here is just an example of one specific gene, and we're looking just for simplicity at 5 cells. So what you can see here is that there is a specific cell that has an alternative start site. So all the other cells, they tend to start the transcript at this first exon. This specific cell starts the transcript at the second exon. It is a known variant of this gene, but again, this information would have been lost if you were only sequencing the 3' end of the transcript just for the identification of this gene being expressed. So by doing full-length sequencing, we can gain a lot of extra information that can be important for the biology of your specific sample. And in the same line, we can also look at polymorphism between the cells and understand kind of if there are any variants that are expressed in specific cells. And again, if you have full-length information, you can get this detail on the full gene body of the gene, not focusing only on variants that may be present only at the 3' end, which will give you a kind of incomplete picture. And again, what this technology allows to do is to be able, for example, to identify gene fusions. So how can we apply this kind of in the immune cells? So we have done several experiments where we wanted, for example, to identify immune infiltrating cells, for example, in a tumor. So we use a kind of a standard kind of tumor line that we were kind of separating between the CD45 positive and CD45 to identify only CD45 positive cells. We went to the process and this analysis, and what we get at the end is something that looks like this. This is a UMAP plot where you can see we can separate the CD45 positive cells between the CD45 negative. But as you can see, even between the CD45 positive cells and the CD45 negative, there are different population of cells of different cell types or cell states that are present in this sample. And by looking at markers, we can then start understanding, all of this is done within our software. You can start identifying what these populations are. So for example, this group of cells are T cells, this group of cells are myeloid cells, this group of cells are neutrophils, and then we can start looking at the kind of CD45 negative, so endothelial cells, tumor cells, and the final one is fibroblasts that were present in the sample. So as you can see, by kind of doing this unbiased analysis, we can look at each individual cell, separate the different classes, so different population of cells, and then looking at specific marker, we can identify what these cells are. And again, at the end, we get this final map, which is the final kind of representation of all the cells and each individual cells, what they are. Another way of using this full-length sequencing is to do T-cell receptor identification. So in this experiment, we use fresh PBMCs that were fact sorted and again went into our workflow, and then we went into Illumina sequencer for doing the library preparation and the sequencing, and then we went to our workflow for doing the data analysis. Again, using a system similar to what I showed before, we can identify the different cell populations that are present in this PBMC sample using markers. But what we can then start to do is to characterize and identify the T-cell receptor repertoire of these cells. And this is what you can see here. You can see that in around 65% of the cells, 55% of the cells, we identified a specific TCR sequence. What again is important to understand here is that for each cell, this is the characteristic of each cell. So 14% of the cells and only have alpha clonotype, around 8% have alpha beta clonotype, and for others they have kind of a variety. And this is very similar to other experiments that have been done on T cells, where there is this kind of variety and complexity of T-cell receptor. Of course, being PBMC cells, a lot of cells are not T cells, so will not express a T-cell receptor. Other kind of type of studies that we can do is, for example, looking at T-cell stimulation and see how kind of these affect the T cells. And as you can see here, we can see that upon stimulation, we can see a marked increase of expression of genes. So as you would expect, there is a significant increase in the number of genes that are expressed upon stimulation. Another way of kind of using this technology is to focus specifically on the TCR. What I've shown you until now is doing kind of a whole transcriptome analysis. In some experiment, what you want to do is really to focus only on the T-cell receptor to look and to study the repertoire of the cells and do kind of a gene expression complementary experiment that can be done from the same library. So for this, we have a specific workflow and a specific kit where we generate the library, and then we separate this cDNA library into two sub-libraries, one to analyze the T-cell receptor and one to analyze the transcriptome. And again, here we're looking at PBMC cells or T cells, where we can see that we have a high kind of clonotype call. As you can show before, there is a variety of the alpha or beta chain that are expressed in the PBMC or in T cells. And we also have a kind of a very high and call of the clonotype. So in 75% of the T cells, we are able to call a full clonotype. And what we can again do is looking at the gene expression side of the library. And for example, in a PBMC cell, we can see again the T cell, B cell, monocyte, and dendritic cells that are expressed into these cells. So just very briefly, as I mentioned before, the system allows to have a complete end-to-end solution. So we also have data analysis packages, and these are kind of two major components. One is the analysis pipeline of the system that will do the multiplexing of the barcodes, all the trimming, aligning, and counting, and then will generate kind of reports. And downstream, we also have more kind of desktop-based interface where kind of molecular biologists can look at the data, identify the different populations as I showed before, and do data interpretation, and also generate images that can be used, for example, for publications. I will not go kind of in the interest of time into the details. If you want to have more information about the specifics of how the software works, I can take a question offline. So just to summarize, the isolated single cell system allows to It's an end-to-end solution for single-cell genomic analysis that give you control of your experiments. We generate only the data that you need. We'll give you high confidence in the data that you're generating, and it's a very flexible platform that will allow you to design the experiment specific for your needs, and also to have an open platform that you can use for developing multiple applications. And that's good science. Thank you for listening, and I'm happy to take any question.
Thank you very much, Daniel, for this so informative presentation concerning KSL's 8 and this ability for single cells analysis, including genomic analysis. So I think that in the future, it will find more and more space in the practice and everyday management of our patients. So here in the hall, do you have some questions? No, I think that everything was so nicely presented. I'm sure that the audience, online participants, were able also to, to join this presentation. So thank you so much. And again, thank you for collaboration and for sponsorship of our conference. Stay safe, stay safe.
Thank you very much, and enjoy the rest of your conference.
Thank you so much.